All right, so it's just about 12:05, so we'll get started. So just a couple of housekeeping things. If you have questions, either try speaking to the mic or speak loudly, and I'll probably repeat them so that the recording picks it up. And if you do have questions during, I usually like this to be more conversational than a lecture, so feel free to raise your hands, stop me, ask. And lastly, all the references that are on the slides, you don't have to spend a lot of time now trying to get them down because these will be made available to you eventually. So all the links and the references will be in there. Okay. So that's me, Vinai Roopchansingh. I'm a scientist for the functional MRI facility. And compared to the other lectures you've had, I think today, if I can be kind to Monty Python and borrow something from them, this is for something completely different. Before we were talking about kind of historical big picture overviews of the field of MR and functional MR and developments. Today we're really going to get down to the nuts and bolts of how the things that we use, these tools, how they work, and some of the math and the physics behind the imaging that we do every day. Okay, so this is the outline of what we'll go through. First thing, I'll talk a bit about an MR scanner, which you see going into a suite, kind of what the pieces are. And then we'll go through, how these pieces actually fit into the acronym, what MRI stands for, and how these pieces actually contribute to the name and why they do. After that, we'll talk a little bit about different types of tissue contrast, why the images that you acquire, how they look, why they look the way they look. And then at the end, we'll go back a little bit, talk about the system again, and just add a little bit more, a few more points now that we've gone through all the material. Okay. So first, our system overview. Okay. So schematically, this is what an MR suite looks like. When you enter one of our suites, you probably walk right into the control room area. So there'll be the computer controlling the scanner where you set up the scanning protocols. You may have some other computers for your experiments. And from this area, you typically have access into the scanner room. And there will be a rather large dressed up magnet. The whole point of that piece of equipment is that it gives you a very strong, very uniform magnetic field, usually to the parts per million is what's needed for the kind of experiments and the work that we do. And around that, very closely, there's a very substantial electronics bay. This provides a lot of the support equipment that drives the scanner, that drives the experiments. So for the first portions of the lecture, besides the magnet, we want to be concerned about the RF subsystem, the radio frequency subsystem. So this is very high power, very high fidelity electronics. We'll explain why that's needed. And then related to the imaging process are gradient amplifiers. And basically what these are very high powered direct current sources that will essentially let us modulate the magnetic field. And again, we'll talk about why we need that. Okay. So what is the magnet for in MRI? So it turns out that some atoms, the nuclei of some atoms, actually behave like microscopic magnets themselves. So the two groups back in the late 30s, so almost 80 years ago now, discovered that certain nuclei with properties where they have either odd numbers of protons or neutrons can behave like little magnets themselves. And this was worth a Nobel Prize back in 1952. So the common nuclei that have this property where the nuclei can behave like little magnets include carbon-13, sodium-23, phosphorus-31, xenon-129. So these are all imaged relatively routinely, but not so common. So does anyone know what is the most common species that we image probably upwards of 99% of the time? So it's actually the simple but mighty proton. So why this is important to us is, one, its abundance in everything, in almost every single molecule that's in your body. Fat, water. So protons from all these different molecules can actually contribute to the signal that we're detecting and to the images that we're trying to generate. Okay? So naturally speaking, if we were to look at the protons of us sitting here in this room at a microscopic scale, they're kind of randomly aligned. Okay? It's just wherever motion diffusion takes them in alignment, the little magnets in them point off in random directions. But if you put yourself or a sample containing protons in a very strong magnet, like we have with an MR system, they tend to line up and have their own little net magnetization. So are people familiar with the concept of taking strong magnet, putting a little bit of iron next to it, and then the iron itself becoming magnetized? And then this in turn can go pick up or attract other things itself. Same idea here, except it's at a much weaker scale, and now we're looking just at the nuclei. But the magnets that we're using are so strong that when you put something containing these magnetizable nuclei into the field, they create a tiny little net magnetization itself. Okay. And just for convention, keep in mind that when we talk about how these magnets are aligned, this is along the z direction. So if you're thinking in 3D space of x, y, and z, you normally align the magnetic field along z. And what happens is, as they're aligned, because they're molecules and they're vibrating and they're tumbling around, even though they have this net magnetization along the magnetic field, it turns out that they're actually rotating and precessing around the magnetic field. So even though the magnetic field is here, you will have essentially the little bar magnet that is the nucleus of these atoms precessing around that magnet, that large magnet. And schematically, that looks something like this. Okay? You can see the magnetic field is along Z, but then the little bar magnet that is the nucleus is kind of rotating around that field. Okay. And here's one of the two equations you should try to keep in mind. It turns out that the frequency at which that nucleus precesses around the main magnetic field is related by this constant called its gyromagnetic ratio, and then the strength of the main magnetic field itself. Okay. This constant is different for any given nuclei. So for protons, it's about 42 megahertz per Tesla. So it means for every Tesla of field that a proton is exposed to, it precesses around at 42 megahertz. Okay. And then other species like that we talked about before, like C13, P31, they have different ratios, but not quite as large. Okay? All right. So this is kind of what the magnet is doing. It's essentially creating this other little magnet that's now in the sample, and it essentially is creating something for us that we can try to detect with the other parts of the experiment. Okay. So this is where the resonance part of the experiment comes in. Okay. So it turns out, now remember we have this large, strong magnet, and now we've taken the protons in our sample and we've lined them up, and they're parallel to that main field. But in this state, we can't actually detect something that's parallel to the main magnetic field. So what you want to do is you actually want to essentially try to tip that net small bar magnet into the perpendicular plane. So if your magnet is going along Z, you want to try to tip that somehow into some piece of this into this plane. And how we do that is by resonance. So this is where the resonance part of the experiment comes in. So if you look at what's happening to this magnet as this, so this now is going to simulate the the RF field. So this is where we inject energy into the system to kind of tip that magnetization away from its alignment with the main field. You can see as it's pointing here, you're hitting the tip, and as it's down, as the magnet's pointing here, you're hitting the trough of the magnetization. So in essence, as this little molecule is rotating around the field, you're essentially injecting energy at that same rate. Okay? And this is very analogous to how, pushing somebody on a swing. Like, as they come to you, you give a little push, and then they go a little further. Whereas if you're pushing randomly, that's not quite as effective or as efficient. So it's the very same idea here with the resonance phenomenon. We try to essentially match the energy at the rate at which it's precessing around the magnetic field by how we apply that energy, so we can move it out of that parallel alignment into perpendicular. And that's how we can detect the signal now. Once it's perpendicular to the main magnetic field, we can actually now try to do some things so we can see it. Okay. So this first bit here of where we can take that magnetization that was parallel and now make it perpendicular, that injection of energy is essentially flipping the magnetization. So a lot of times you'll hear about a flip angle in terms of what you use for your experiment. Well, that's this bit of this part of the experiment. The flip angle tells you how far away that you've essentially rotated your magnetization down. Okay, and this is one type of contrast we can use to determine different, bring out the differences between different types of tissue. And even if, and then even how we apply the RF can also bring out different subtle things in the tissue. So again, going back to the swing analogy, if a person comes to you and you give one big push, they go pretty far out. Whereas you can do the same thing where every time they come to you, you can give them a smaller push, and the net effect is still the same. It takes longer, but it kind of gives you the same effect. And all these little subtle differences can bring out different properties of the tissue. Okay. So now we have the magnet, we have resonance, but because our magnetic field is so uniform, there's no way for us to actually localize what's happening with the signal once we've excited it, once it's excited and generating. Okay? So we have a hard time localizing it. So this is, start talking a little bit about imaging. So remember, uniform magnetic field, it's parts per million. So if you inject your RF and then are listening back for what the proton is doing, from all over the volume, it kind of looks the same. Ideally, it should look the same. Okay. And how we get around that, how we try to apply some spatial localization, is we actually do some very controlled distortions to the main magnetic field. So again, we start off with a uniform magnet, again, uniform to parts per million. And to get the protons from here to do slightly different things than the protons in the other part of the volume, we distort the field in a very controlled way that we know. Okay? And schematically, you have a magnet, and you have this nice uniform imaging volume. And when we actually want to image, what we do is we take that field and we apply these gradients to it. Okay? The main magnetic field is on the order of Tesla. We usually measure our field distortions in millitesla, so tenths of a percent. So much smaller in terms of scale, but it's enough for us to essentially differentiate protons now in this part of the volume versus protons in that part of the volume. Okay. This is essentially saying that. So remember our original equation where we had our frequency at which the protons are precessing or spinning is dependent on the field that they see. Now, if we change that field according to its position, now the frequency at which these protons are precessing and behaving also will vary as a function of position. And since this is essentially an imposition on the main magnetic field, it can be represented this way, mathematically. pictorially, this is what that would end up being. So again, we have our main magnetic field, and if we apply a gradient along the z direction, then at this point where the gradient is a little bit higher, you would have one frequency, and then that bit of tissue will have protons precessing at omega 1, which is a little bit higher than protons in this slab here, which would be precessing at omega 2, similarly omega 3. Okay. And here, if we're trying to image Homer's brain, what frequency would we need here to actually excite and then detect that? Yeah, exactly. This one here, because the bulk of the brain is at 64.007 megahertz. If we were to excite at 6405, completely miss that, maybe get a little bit of his eyeball, and then 009 is off here, where in Homer's head it's empty space. Okay. So this is basically one dimension of using your gradients to do encoding. Okay. What we do is we would apply a gradient, and if we want to get Homer's brain, the transmit frequency that we would use to essentially resonate with the protons in this bit of tissue here would be at this frequency, 64.007 megahertz. Okay. And if Homer had a slightly more substantial brain and we wanted to get a different slab, then we would excite the slab here. So this is actually what happens normally in our subjects. When you do a 2D slice excitation experiment, you get your different slices by essentially having this gradient on and then transmitting very slightly different frequencies that resonates with the tissue in that at that space. Okay. So this is what we call slice excitation. Okay. But we can use the same idea of essentially varying our fields, again linearly, but in our other dimensions, to continue help us with the localization process. Okay. So the first dimension is called frequency encoding. The second is called phase encoding. And I don't have a picture for this one, but I'll describe it as we work through the next. So now we've gotten our slab of tissue in Z. If we apply a gradient now from Homer's left side to right side, we again now have a variance in frequencies across that axis. Okay, so now protons in this slab of tissue have been excited, and they're all precessing. Now, as you apply a gradient along this direction, protons on this side will precess at slightly slower frequencies than protons on this side here. And it turns out that when you do this readout, you're reading all of these out together and aggregating it. Okay. And you end up with a signal that looks something like this. It turns out there's a little bit of math called Fourier theory. It says if you have any arbitrary shape, image, signal, you can take anything arbitrary or random, and if you have enough terms that vary regularly, the most common sets are sines and cosines, so things that vary periodically, like that RF waveform showing in some earlier videos, or even these waveforms here. If you have enough of these at high enough frequencies, i.e. varying very quickly, you can represent almost anything with essentially a combination of these waveforms. Okay, and it turns out that's kind of what we're doing with our gradients and the imaging process in MR. Okay, as we turn in our gradients, we're applying this kind of encoding of all these regularly varying waveforms. And if we look at what our gradients are doing over time, they're filling out this mathematical construct called k-space. Okay. So people here been down at any of the scanner suites at all? Have people looked at like the oscilloscopes that we have at some of our scanners, seen them on and seen waveforms as the scanner is operating? Okay. If you have a chance to do that. We don't have it on our newest scanners right now because we need to buy some interface equipment to replicate that. But if you look at what the waveforms are doing, this is essentially drawing out how it's creating and walking through k-space. Okay. So what k-space represents mathematically is essentially the time integral of the area of the gradients. Okay. So let's walk through these 5 points here pictorially with this really simple experiment. So first off, you turn on a gradient where you want to select your slab of tissue, your slices, and you have your RF that resonates with this whatever frequency you create at that location. Okay, so you're still right. These 2 points actually leave you at the center of k-space. So the starting point of k-space is kind of equilibrium, where you put your sample there, things are laying down, like at rest. You haven't applied any RF, you haven't put on any gradients. It's just kind of your equilibrium state just laying there in the magnet. Okay. So when you excite with your slice selection and then your RF pulse, you don't move away from k-space in this XYZ, or sorry, KX, KYZ plane. But now as you turn on those gradients, so now after B, so A and B are still at the center here, as you turn on your Y gradient, it takes you out to this corner of k-space here, and your X gradient is negative, so that actually takes you back in negative X. So KX is positive this way, KY is positive this way. So positive KY takes you up, negative KY takes you this way, to your left. And now we get to this point here, and this is where you are at point C. And then as we turn on our X gradient here now, so we create our range of frequencies that we read out, now that will actually walk us from one side of k-space along kx to the opposite side. And almost all imaging involves trying to fill out k-space. Here it's in a Cartesian pattern, but, and this is still the majority of what we do, but there are some more interesting ways that you can essentially accomplish acquiring this, acquiring the data that fills you out, depending on what you do with your gradients. Okay. A couple things to keep in mind now. So remember, this is you're going to acquire this data and essentially do a transform on this to get your image. Okay. In this domain, this spacing here actually is what determines your field of view. So if you have a small spacing here, it turns out you have a very big field of view. And if you have a large spacing here, it means your field of view is very small. And how far you go out in kx and ky determines your resolution in those dimensions. So if you go super far out in kx, then means you have a very high resolution in your x direction. And if you go super far out in ky, same for that direction. Okay. So one thing to take away from this is that even though we do these things cohesively and most of our images turn out to be square, we can manipulate these things on a per-dimensional basis independently. Okay. All right. And physically, so the first time that I was trying to get this to gel and to have some intuition of what it means, this was one of the pictures that actually helped me kind of come to the realization of what all this means. So again, if we picture this as our k-space, So essentially this grid here. Okay. If we walk along the x direction here, it means that we're encoding at this location here something that has this variation. We're just varying, we have a sinusoidal variation along this way, right? If we go straight up in the y direction, we have something that varies along y sinusoidally. So again, it's sinusoidal, but now the variation is this way. Sorry. Y. So that's what this here represents when you are back in image space. And if you look at what variation or what waveform B3 represents, this is now has some variation both in x and in y. So now it's a kind of a diagonal pattern. As you apply your gradients and your encoding, you're essentially combining things that have these kind of variations, these sinusoidal variations, across this grid that you're acquiring. People familiar with graphic equalizers, audio equalizers at all? Where you can essentially bring out different aspects of the music depending on its frequency. If you want your bass, you bring up your lower frequencies. If you want more treble, higher frequencies. So very similar idea to this, where whatever waveform you're trying to recreate, you're balancing how much of each frequency you have to add to give you back your image. Okay. And that's all kind of formalized in this equation, which is the Fourier transform. Hi, yes?
Could you go back a slide? Can I ask a clarifying question?
Absolutely.
So in figure A, the three on the left are increasing in frequency, right? So, the frequency is getting higher with like the third one, an A, than it is in the top left, right?
Yes.
Okay, but so how is that on Kx or Ky? Where is like that frequency being encoded in K space?
Okay. So the question was, in this diagram, how do A and B correspond? So if we were just looking at variations along, so let's just look at this along the x dimension. A would probably be somewhere here, B would probably be somewhere here, and C would be somewhere here, so as we go across. So it just means you're getting out to higher and higher frequencies along your horizontal dimension.
So it's Kx is essentially encoding the frequency, and then Ky is encoding the direction of the waveform?
So that's actually encoding another property of the waveform called its phase. So because these things have both an amplitude and a direction, one dimension encodes how fast it's going, and the other direction encodes the actual position that it ends up as. If you actually go into the math, these end up actually being equivalent things. So it's kind of arbitrary how we name the encodings, but it was kind of a simplification in the beginning to kind of differentiate the two dimensions and how they were detected, essentially.
Thank you.
Okay, so that's formally represented by this equation here. So there is an integral over all space. So the signal at any given time is essentially the integral over all space of the magnetization over that same space. And this kt here represents the time integral of what's happening with your gradients at that time. Okay, and as it turns out conveniently, this here is exactly a Fourier transform relationship between where we are kx and position. So if we invert this with the signal that we detect, we essentially get back an image in x, y, and z. So the Fourier transform here, pair here, is between k-space, which is a function of time, and then position. Okay, so it's a little bit abstract, but that's kind of where this is how this is formalized. So just to give you a few more concrete examples, if we take this this portrait of Abraham Lincoln and put it into Fourier space, Okay, you get this. Most of the images that we get, our K-space does not have this distinctive crosshair pattern, but it does have the general characteristic that it's brighter in the middle, there's a lot of energy there, and it's more diffuse as you go further out. Okay, so remember I was saying before, as you're in the middle here, closer to the middle, you encode things that vary more slowly, that have lower spatial frequencies. And as you go out further here, these are things that vary more quickly over space, and those encode higher detail. So that's shown in this set of pictures here. So if you take that same Fourier transform, if you cut out the middle, you cut out all the slow varying stuff, and you cut out a lot of the energy, what's left behind actually just gives you the edges of the image. Okay. And if you do the inverse, where you take out the edges and you just are left with the middle, it still has a lot of the energy of the picture, but now the edges are not anywhere near as clear as they are if you had the whole thing. Okay. So there's always a balance in terms of, okay, you want to get as much K-space as you can, you want to get it as densely as you can. So a dense K-space where your points are close together means you have a big field of view, and then if you want resolution, then you need to go far out. But in order to travel this way, your gradients need to be on at higher amplitudes and for longer times, which requires more energy. Okay. So now you can maybe start getting to see why some of these things may be limited, because gradients are limited in how far we can push them, so then you have to spend more time if you want to get more resolution. So in order to get something at the appropriate resolution and field of view for experiments, we always have to play a balance in terms of what our scanners can do versus what kind of K-space coverage you need to get that. Okay. So a couple of the trajectories that are most common for imaging, for high performance imaging, are echoplanar and spiral. And this here is an EPI trajectory. Similarly to the example we had a few slides ago, you start off in the middle. You have a couple of essentially what's called pre-winder gradients that get you off to the corner. Then you turn on your readout gradient here, turn it off here, add a little phase encode, turn back on your readout gradient here, turn it off, phase encode, and you do that over and over again till you get through K-space. In order to do this, what you're doing here is you're turning on your gradients maximum polarity, maximal amplitude at one polarity. And then when you turn them off, then you quickly switch them maximum amplitude to the opposite polarity. And this is very physically and electrically demanding on the systems. Okay, spiral, this doesn't have the hard turns that EPI does. These are more, so I'll have examples of what the waveforms are in the next slides. These are more like sinusoids that keep increasing in frequency, but still very demanding in terms of timing and power requirements. Okay, so if our scopes are on, the next time you go into a suite, you should see something resembling this very closely for EPI. There's not a lot of spiral done here, but there are a couple of sequences that have that embedded. So those are the kind of waveforms you should see. Okay, so that's kind of the bits of the acronym. We've gotten to the imaging. Are there any questions here? This is kind of a lot of concepts to digest. Yes?
So why would you use one or the other, ways to travel in space?
Okay, so the question was, why would you use one method of traveling through K-space versus another? Essentially, the effects that these have on the data on your final image quality. So one thing that we need for a lot of functional MRI is actually bold contrast. And you need some amount of time for that to evolve. Okay. For spiral imaging, if you start here, you essentially, there's a lot of dead time where your scanner is not doing anything, that's kind of wasted for collecting data. So whereas with echoplanar, if you time your experiment right, by the time you've collected this data and get to the center of K-space, which is where the bulk of your bowl contrast is encoded, then your time overhead is not as high. Your time penalty is not as high. You don't have as much dead time in your sequence. You can still spend that 15 milliseconds or so acquiring data. Whereas here, if you want to start acquiring data with your bowl contrast, then you have at 3T, you have about 25 to 30 milliseconds of dead time where you're not doing anything. These echoplanar images tend to be have different distortion characteristics. So echoplanar images tend to be more distorted along the phase encode direction. So not along the direction that you're traveling this way, but like where these little blips, where you're jumping from essentially KY line to KY line, the distortions tend to be more prominent along this direction, because this walking down this way is a slightly slower process than this way. Whereas spiral scans tend to be more blurred uniformly. Spiral, although it's gotten better, but traditionally it was more sensitive to being suffering image artifacts from off-resonance effects. So it really does depend on the experiments and how well you can essentially control for the weaknesses of each of the sequences. So, any other? Yes.
How does the time moving through K-space relate to your TMR? Does it?
So the time that you spend collecting K-space is, so I can speak practically for what most people are doing right now. So the time to acquire, say, a 3 millimeter echoplanar scan is on the order of maybe 30 to 40 milliseconds right now at our 3 Tesla scanners. Okay, but that is one slice worth of data. And then you have to spend a little bit more time with encoding. So say you have 50 milliseconds per slice that you need. So with a 2-second TR, how many slices does that limit you to? So about 40 slices. Right, so 50 milliseconds times 42 seconds. Right, so the effort that you have to expend collecting K-space, this kind of is what you have to make sure that you have enough time to do in your TR. Yes?
What's the meaning of giving 3 different gradients on 3 different axes?
So the question was the meaning of the different gradients on the different axes. This is all the different dimensions that we're encoding our signal. So this is a 2D echoplanar experiment. So what we're doing here is we are exciting here at a frequency that we set with our gradient. Okay, but because, remember I was saying that the effect of the gradients, we are conveniently labeling them as slice select, phase encode, and frequency encode. But it turns out that if you actually look at their effects in K-space, this is actually also doing a little bit of encoding in KZ with our RF that we unwind and essentially bring back to the middle of K-space. And then what we do with our gradients here is we move these off in the different dimensions of K-space and walk through and acquire when we need to acquire. So essentially the role of the gradients are to move your signal, your magnetization through K-space.
So for certain experiment, you can give same gradients to 3 different axes.
Yes, the question was in terms of different experiments, you can have experiments where you have the same gradients on different axes, and yes, absolutely. This is one of the things that we'll talk about in the next section is with MR, because you're imaging essentially the bulk effects of things you're imaging at the macroscopic level things from essentially the microscopic / subatomic level. How you play gradients, and even just if you have the same things, if you just time them differently, you can really get differences in contrast and how the tissue looks in the image that you end up with. And that's really one of the nice things about MR. Okay. So we'll get into some tissue contrast, unless there are more questions about what we just went through. Okay. All right. So images like this should be fairly, probably, most people seen images like this? Okay. This is an MP Rage, it's a spin echo type of image. But what we are actually looking at here is the response of the protons over time into different ways of essentially encoding the signal and then detecting them after different amounts of time. Okay. So there are two basic types of contrast that we utilize in MR. And there are lots of others, and there'll be lots of other talks. We'll talk about those. But the two basic ones are what's called T1 and T2. Okay. So T1 is a reflection of how long the signal takes to actually get back to equilibrium. So remember, we started off with our patient, our animal, our sample. We put it in the magnet, and it takes us a couple minutes to kind of set things up. And by that time, equilibrium has been established, and then we come outside and we start imaging. And once we start imaging, for the imaging process, we perturb that magnetization away from equilibrium. But to be able to continue imaging, we depend on that equilibrium signal. So T1 is actually a measure of how long it takes for the signal, the magnetization of the sample to regrow. Okay. So, and if you look at how the vector along here plays out, essentially T1 is a measure of how long that takes to get back. So essentially something with a longer T1 grows back much more slowly, and something with a shorter T1 recovers very quickly. And why this is important is because I think people realize now with MR, we're doing things over and over and over and over again. It's rare that we do things once and we're done. The experiments, a lot of the encoding, a lot of experiments we do, even just some of the basic imaging, you have to repeat certain parts of the process over and over again, varying things a little bit each time. And they all depend on having signal available to you to detect. And the amount of signal that you detect is primarily determined by the T1 of the tissue. If you have tissue with a really long T1, then you have to wait a longer time for it to recover to get back signal that's useful to you. Okay, so that's one of the reasons where TR comes into play. If you have tissue with a long T1 and you want to keep imaging reasonably from it, then you need a longer TR. Okay. If you have tissues with shorter T1, then you can image much more quickly. This is one of, so have people image here at our 3Ts and our 7Ts? Okay, so if you notice that your T1 weighted images are actually longer at 7T than at 3T. And that's exactly because of this effect. Because at 7T, it actually takes longer for this equilibrium signal to recover than it does at 3T for the image, especially for brain tissue. Okay, and these are the kinds of these are the kinds of images that we get from a T1 weighted image. So in this signal here, the white matter has a relatively short T1, so it's brighter by the time we image, and the gray matter has a longer T1, so it's still a little bit darker at the parameters that we're using for this experiment. Okay. This particular imaging type, we use CSF suppression, so that's why you don't see the cerebrospinal fluid here. But that also has a relatively long T1, so that should have been pretty, so it would be relatively dark in these images as well. So that's the recovery back to equilibrium. The second type of contrast that we utilize determines how well all the nuclei in a given sample maintain coherence with each other. So we're looking at millions and billions of nuclei. And at the beginning of this experiment, they're nice and aligned, and they have a net signal that we can actually detect. But remember, just as we're sitting in this room here, things are kind of randomly aligned. And that's where things kind of like to be, random. And this is where, after we excite, the transverse signal will tend back to. So after we excite, all our nuclei, the magnetization from all these nuclei, nominally point in the same direction. Okay, and that gives you a nice long vector here for a signal, a strong signal that we can detect. But over time, as they keep processing, you can see they're kind of getting out of step with each other. And the net signal that they have, which is kind of the net addition of all these things added up, shrinks away. Okay. So this mechanism here, where you essentially look at the coherence of the spins with each other, this is called T2. Okay. And this is a kind of image that you use that's giving you T2 weighted contrast. So fluid here and here, nice and bright, longer T2, and followed by gray matter, which has slightly shorter T2, and then white matter here, which has a pretty short T2. Questions? Yes?
When you say it has a longer T2, are you saying that it takes longer for them to get out of sync with each other, or it's...
So yes, a longer T2 does mean it takes longer for the signal to decay away. Yeah.
Thank you.
And you're saying this depends on the tissue, right? What aspects of the tissue?
What affects the T2 of a tissue composition, like complexity of, like if there's a lot of protein molecules in there, metabolites, electrolytes.
Something
Yeah.
magnetic, basically.
No, actually it's not. It's tissue composition. And it doesn't have to be anything specifically magnetic per se. All right, and then the last bit of contrast I'll talk about is bowl contrast, which is not these maps that people typically see with bowl contrast. Bowl contrast is much more subtle than either T1 or T2. So a lot of times when people show maps like this of bold activation, these are statistically derived parameter maps that are derived from essentially the time series of the experiments. So where bowl comes from is very much at the macroscopic level and very much from a T2 type of derived mechanism. Okay, so if you have a blood vessel in a section of gray matter that's active for your experiment, or let's start off with it at rest. So in this example here. Okay, so this is a cross section of a capillary, and then this is gray matter tissue around it. At normal levels, you expect if this hunk of tissue is not doing anything intensive, brain tissue is not doing anything intensive, you expect that there's some amount of oxygen extracted from the blood in this capillary, and the result of that is that there's a bit more deoxyhemoglobin. Okay. Turns out that there's a slight difference in magnetic properties between oxyhemoglobin and deoxyhemoglobin. When that oxygen molecule gets sucked up into the tissue, for metabolism, whatever else is going on, that heme core has some iron in it that's more exposed, and that actually it creates a slightly stronger local magnetic field, and that's what's showing here. So out here in the surrounding tissue, where there's water, there's not a lot of molecules or species that are creating these stronger magnetic fields. The field lines are further apart. Whereas in the blood vessel here, where there's some amount of deoxyhemoglobin, this is a little bit more magnetic. So the field lines here are closer. And there is now a little local microscopic gradient in magnetic field between the inside of the blood vessel and the outside. Okay. And if you think back to your T2 mechanism where things are getting out of phase depending on how they interact with each other, this little local field gradient essentially amplifies that effect. So something in here, because this is a slightly higher magnetic field, will precess slightly faster than something outside here. And that result is that there are thousands. So this is on the order of microns. Okay, our imaging volumes with MR right now are on the order of millimeters. So there are thousands of these in any given voxel that we image. Okay, so the net effect of all these little microscopic gradients is your signal decays away relatively quickly. All right. If now you have your subject or your animal do something where you engage the surrounding bit of cortex, okay, the bold, what we've come to learn is now you have an oversupply of oxygen and oxyhemoglobin, contrary to what people first thought the effect would be. And it turns out now, because you have more oxyhemoglobin, less deoxyhemoglobin, now the magnetic field in the blood vessel actually becomes slightly weaker and more analogous to what's outside. Okay? The net result of this is that now there is less dephasing between signals out, protons precessing out here and protons precessing in here. So that when you detect the signal, you detect a higher net signal. Okay? So if you're doing an experimental design where you have off time and on time where you're engaging that slab of cortex, your net signal statistically in that slab of tissue is going to be higher. And this statistical correlation and other related things are how you derive these parameter maps. Okay? And because you start off with kind of looking at how spins interact with each other, but now you have the additional complication of the background gradient, this is actually called a T2 star contrast mechanism. Okay. And so I lumped this in with a bunch of others, but are there any questions before I run through some of these final bits of? Okay. So that mechanism there, that is the basis of BOLD. It's blood oxygen level dependent contrast, and it comes from the fact that when that bit of tissue becomes active, now your oxygenation level goes up, and essentially that causes the signal to go up as well. Okay, all right. In addition to the basic types of contrast that I've talked about here, depending on how you manipulate RF pulses, gradients, timing, you can do fluid suppression. So this is the basis of what's flare imaging. This is another type of imaging where tissue contrast, you've knocked out signal from cerebrospinal fluid, but you have less contrast between gray and white matter than you typically do in the T1 related, the T1 weighted example I showed previously. Okay. Magnetization contrast looks at trying to get at the signal that's related to water molecules that might be bound by protein pools. Okay. Perfusion imaging is trying to get at how much blood is flowing to a given volume and it is actually leaving capillaries and entering tissues. Diffusion weighting, which is another one that's pretty heavily used locally, depends on, or actually gives weight to how far water molecules can diffuse away from essentially the start of the imaging experiment. And Jeff Dunn's group a few years ago showed just remarkable, absolutely fantastic looking images, but looking at the phase of magnetization, and they were trying to see now if they can use this clinically. Okay. So, any questions on? So this is kind of the high points in contrast. Said people will have a few more throughout this lecture series, and who knows, hopefully some of you will come up with some new, fun, interesting ways of looking at water and what's in our bodies. Okay. So finally, we have our magnet that essentially generates our signal, or a little bit of magnetization. We have our RF system here that we transmit energy into our system and then detect with. And this is where our resonance comes in. And then we have spatial encoding using the gradient systems. That gives us our 2D and 3D localization of our signal. And all of these things can be modulated with exquisite precision with control systems that will help you, essentially, that will let you bring out different contrasts in tissues and samples. All right, that's about all I had today. Happily take any more questions that people didn't ask during the talk. Thank you.